welcome to the May 15th meeting of the Working Group and GLIMPRINT. Thank you, everybody, for coming at our new time of 10 a.m. We hope that this is going to be more accessible for our European colleagues, and we apologize to anyone on the West Coast of the United States for having to get up early. We have a single talk today from Eran Agmon from University of Connecticut. As usual, I need to remind people that this meeting is live streamed and recorded and available in mildly edited form on YouTube, and so please be aware of that. As always, Tom Helikar, myself, Jim Sluka, and Lorenzo, as the people who organize this and run it, are here to take your comments and suggestions. Please don't hesitate to reach out to any of us if there's anything we can do better. This is really here to build community, and we're always looking for ideas about how we can do that more effectively. We have a variety of communication channels. They work better if people use them. Some of them are quite active, some are less active. I don't know if the QR codes will work. You can try. Our biggest efforts would be the IMAG MSM wiki page, which has lists of products. There's a LinkedIn page, there's a YouTube channel, and there's the GLIMPRINT homepage as well. We'd love to have people volunteer to help us maintain those. At the moment, Jim Sluka does it, and we always could use help with that. If you have a publication to share, we can put that up related to these topics on the IMAG page. Give you a little bit of extra publicity. We're always glad to do that. And there's also the GLIMPRINT page. The GLIMPRINT page also lets you announce seminars, meetings, workshops, publications, job openings, funding, and other things. Again, if you have any trouble with any of these, please email jsluka@iu.edu. He's the keeper of the websites. Coming up, there is a workshop on modeling, optimization, and control in biomedicine. And if you're interested, please try this QR code here. Tom Cho from UCLA will be there. Reinhardt will be there. I wish I could be there, frankly. It looks really good in Frankfurt. And Lucas is the organizer. Thought I saw Lucas on the call. Was there anything you wanted to add if you're here? There you are. Yes, go ahead.
Yes. Thanks, James. Yeah, the workshop is really open for everyone. We also try to make contact with some like colleagues from industry. There are a lot of pharmaceutical companies in the Rhine-Main area and in Basel. So we hope that some of them will also join. And yeah, the event is free of charge. If you're interested, just register.
Great, thank you. That sounds like really glad you're doing that. It's a great opportunity for people. So please reach out to Lucas if you're going to, if you want more information. He's here on the call, so you can direct message him if you like. We have our CompuCell workshop coming up, a virtual workshop at the end of July, beginning of August. If you'd like to have you or your students participate in learning how to use CompuCell 3D for multicell modeling, we'd be delighted to have you. This year there's a $25 registration fee, but it's fundamentally free. We'd love to have people participate. If you happen to have used CompuCell to do this kind of modeling and you'd like to present at that workshop, we'd love to have you as well, so let us know. This summer, the deadline for submissions is passed, but participation is still available. There's going to be a model sharing workshop at the Society for Mathematical Biology in Edmonton this summer. Registration is still open for that. There are two things. There are going to be a series of mini symposia, both on model sharing and GLIMPRINT on digital twins. Those are going to have 12 speakers each during the regular meeting, and then the day before the regular meeting starts, there's going to be a satellite meeting at the university on model sharing. And if you'd like to sign up for that or get more information, feel free to reach out to me. I'll be glad to help out. Grace mentions that there is going to be a track at ISMB in July 20th to 24th, which includes digital twins. And so if people are interested in that, the organizer is Fenglo Mao at NIH. The email address is here at the bottom if one wants to find out about that meeting. There's an FDA initiative on novel or new approach methodologies. We've had a lot of talks about those over the past few years. If people are interested, please take a look at that funding opportunity. There's been a lot of movement in the direction of NAMs by funding agencies. Nicole Kleinstrayer is one of the leads on that, and she's presented here in the past. Please reach out to Nicole or take a look here at the FDA list if you're on the FDA side rather than the NIH side as well. Coming up, we have a pretty full schedule. We have 2 speakers from Indianapolis coming up. We have a talk on compartment model-informed neural networks. We have Jacob Barak, who was very active in this group early on, who's going to try to take us back to our roots and present some materials and ideas about the COVID epidemic and what we've learned from it. That was how things got started 5 years ago, and so that could be quite interesting. We weren't planning on having many meetings in August since people tend to take vacations, but there are some open slots in August. We have plenty of slots open in the fall. As I've said before, we really try to schedule talks that are of interest to the community. We realize it's a big community and a varied community. So please suggest yourself, your students, your postdocs, if you have something that is relevant to anything broadly in the area of multiscale modeling, immunology, quantitative biology, translation, bioengineering. If you've read a paper that you think is interesting and you'd like to hear the speaker from the paper, let us know. Lorenzo Veschiti is extremely good at getting people to come to speak. And if you've spoken yourself, don't be shy if it's been a year or two to come back. Even if it's been less than a year or two and you've done other work that you'd like to present, please do feel free to suggest yourself. We're always looking for speakers. And again, the reason this seminar series exists is to help build community and giving opportunities also to students. You don't have to be a senior researcher to present here. We will have a steering committee meeting tomorrow at 11 a.m. EDT as usual. If you think you should be invited, let us know. We're happy to invite you. If you come for one steering committee meeting and don't come back, that's all right. But we typically have the steering committee's meetings the week of the seminars to discuss projects, opportunities. We've been talking about combining this seminar series with one sponsored by Virtual Human Development at Toronto, led by Maria Abu-Chakra. And so we may discuss that again tomorrow, whether we would be able to find a way of connecting those 2 projects together. There's also been an initiative from Europe to create a network of researchers in the area of modeling the liver and liver toxicology in response to disease and treatment. And we may discuss also what could be involved in supporting that initiative of building a worldwide network on liver modeling. Today, we're very pleased to have Eran Agmon from University of Connecticut coming to talk to us about his work on compositional systems biology with Vivarium and some of his work on E. coli modeling, enabling scalable hybrid simulations with cellular systems. This is a topic that we have discussed a lot over the years in a variety of forms. We're very excited to have Eran come and present to us today.
Thanks for having me. I'm Eran Agmon from the UConn School of Medicine and the Center for Cell Analysis and Modeling. So let's get started. Well, you might ask, what is compositional systems biology? no, this is not computational systems biology. This is a new field with one researcher in it, and that's me. So if you haven't heard of this before, it's because I just made it up. So to make a contrast between what I'm going to call compositional systems biology and traditional systems biology, whereas traditional systems biology might focus on the structure or dynamics of specific subsystems and controlled conditions, and you can see different mathematical frameworks for describing these different subsystems. There's different fields have launched in biology, and mathematics have been found that best describe those particular subsystems. Of course, there's many ways to describe them, but here we see metabolism with flux balance analysis, morphogen gradients, maybe some PDEs. We have agent-based models for multicellularity, maybe some ODEs, you name it, missing rule-based models. So in compositional systems biology, we're more interested in how to connect them. We're interested in the space between models. I'm trying to do like a figure ground reversal for you, in which it's more about the connections and how we bring these things together into hybrid models. There's some interesting questions that we can ask about this. Here, maybe we have three models: transcription, translation, metabolism. What variables should these submodels expose through their interfaces? We can try to list them. Different levels of resolution might come in depending on what our needs are. How do coupled models connect? If we have these submodels, how do we bring them together? So here I'm showing that as wiring diagrams. This will become more concrete and formal as the talk goes on. How do models translate across scales? We have a bunch of different molecular models connected. How do they come to exhibit cell scale properties? How do models of cellularity, how do they translate to multicellularity tissues? And can we build infrastructure for the open-ended composition of these models by a community of researchers? I'd be interested in compositional biology is about connecting communities. Can we download models, add to them, share our models together? And of course, questions about standards and protocols come into play in order to support this kind of vision. Okay, a little bit of background. A couple of weeks ago, Marcus Covert presented on the E. coli whole cell model May 1st. If you want to see some more of that, I'm gonna. This was the lab I was in as a postdoc. Marcus was very kind to bring me in and to try my crazy ideas. This was a very large-scale project for E. coli, in which there were several modelers in the lab, experimentalists, all trying to capture different aspects of cell physiology of E. coli, combine many data sets together, and come out with a large simulation that covers as much phenomena as we can, as we can. And it's still a work in progress. We're about to launch a next stage of development for the E. coli project. What you should know about it is that this is a hybrid model. As I said before, different cellular functions require different mathematics. And so we have all kinds of different variables represented in different data structures, mathematical models to represent their processes, and we run them all together as a hybrid system. Repeating these calls, you can think of it kind of like a co-simulation. What I got out of the first couple of years of my time in Marcus's lab was that we need proper software to streamline this kind of integration. This is not something we should do, just open up a Python file and start putting these together. For this to be really at scale, we need software that knows what it's doing to scaffold the kind of models that we want to build. So here, imagine having a multi-scale model crossing from molecular to multicellular networks, go in, and look at the data that informs the different layers, look at the different submodels, maybe suggest a new submodel that you think is going to improve model prediction and plug it in there. Okay, and so here comes Ryan Spangler, software engineer, who just started up at UConn Health, but we've been collaborating for a long time to help us build this software, which is the Vivarium software. I'm going to go a little bit into the formal under the framework underlying Vivarium and basics, and then we're going to go and look at some examples of how this works. The formal framework is based off of, it's an evolutionary extension of Robin Milner's bigraphs. You can see that whereas Milner had these hyper edges as one of the types of graphs, we've replaced them with processes. So for example, E1 becomes a P1. I'll show you the consequences of that. Okay, but first, some basics. So this is the visual language that we've developed for process biographs. You have these rectangles representing processes. These are the models. They take some variables and inputs, run for some time, and submit an output. Stores can hold your different data classes, different data types. Really, any kind of data can be held in the store. You can put them in a hierarchy, wire them together, and then run them through with different orchestration patterns. We'll see all of this come into play in just a moment. Okay, so we're going to start off with the most minimal example I can possibly think of, and stepwise through composition, add additional details until we have something quite interesting. So to start, let's say we have this transcription module. There's an ODE in here, single ODE. It reads the DNA, writes the mRNA. This is the kind of thing that happens when you simulate it. Can then add a translation. Now we just have really two coupled ODEs. We could, in principle, do this in a single module, but for demonstration, I'm separating out to two separate modules. So transcription reads the DNA, writes mRNA, translation reads the mRNA, writes the protein. We have the central dogma going. Inside of here, we just have a single DNA called G, single mRNA called C, and single protein called X. You can see the DNA is unchanging, protein increases. There's a degradation term inside of transcription, which is why this reaches a steady state. And you can see the protein soon after that. Okay, I want to talk a little bit about orchestration patterns. There's different ways to trigger these processes, to call them in certain order. We're going to look at this one on the left, multi-time stepping, in just a second. A temporal process can have a fixed interval, think just Euler method, in which at the start of every time step, it tells the Vivarium simulation engine how long it needs for its time step. Maybe it's a fixed amount. And the engine says, puts it on the scheduler and lets it run. You can also have adaptive time steps. So if a process says it needs shorter or longer, the engine takes that time step, puts it on the scheduler, and runs it. What that lets us do is combine stochastic and deterministic systems. Over here, you can see that I've replaced the transcription process, the ODE, with the stochastic transcription process. And so you can see this kind of stochastic trace over here as the mRNA is expressed. We have an adapter step. So the stochastic model operates on counts, typically small amounts of counts, and the deterministic, the ODE, operates on concentrations. So we need a way to convert between these two different data types, and hence this adapter step. You can see there's a bidirectional arrows here between RNA in concentration format and RNA in counts format. If any one of these is updated, it reads in the volume and triggers an instantaneous update to the other one, so that these are always in sync with each other. But now we can connect one module to the data type that it prefers over here on counts, this translation to the data type it prefers on concentrations. I think I need to explain the adapter step just a little bit more. I showed you these orchestration methods. So we have multi-time stepping. You can also trigger a workflow. This is like a directed acyclic graph. If a state gets updated, it triggers a cascade of updates. Let me show you what I mean by that. So we have the multi-time stepping. Between the time steps, we can run these workflows. So what happens over here is any time that one of these is updated by a temporal process between the time step, before the next time step happens, this converter is triggered. Oh, sorry about that. Okay. There's other ways to combine orchestration methods we might see later. The next thing I want to show you is hierarchical embedding. So we can take what we call a place graph and nest states within it. Over here, we see a cell at the top. We break it down into cytoplasm, going down to complexes, and we might have some individual proteins at the bottom. This is just the static state. Here I'm showing how we can use the hierarchy to connect processes across scales as well. So below over here, this is the same model I showed you before. So notice the DNA and concentrations, mRNA, we have this this translator in there. So it's just this model, but now I've nested it. So we have a model under this 0. We have we have a single cell called 0, and it has the model inside of it. Now I've put additional processes in the environment. We have a multi-body physics engine that operates on the cell bodies and moves them apart, volume exclusion. And we have a diffusion process that connects the fields and the cells, so like a PDE. So think of it like here we have a PDE field and we have a cell body, and inside of that cell we have the previous model that I had shown you. Okay, one more thing about orchestration. Okay, this is the final one over here. We also have event-driven graph rewrite rules passed in. So if a process triggers a graph rewrite rule, you can actually change the structure of our hierarchy. For example, here we might start off with an environment that has a single cell inside. You need a graph rewrite rule for division, and now you have a kind of a new state in which you have 2 cells. One cell might engulf another. You need to move it inside, so that now agent 3, its external state is agent 2, and it lives inside of it like a little parasite. Agent 3 might burst. We have to go back to this one. Its internal contents spill into agent 2. This is how we use the event-driven graph rewrite. For this model, there's a oh, I didn't show the division. There's a division process that I failed to show here. But you can see before division, we have this hierarchy. and it sends a message up to cells when a threshold is passed, and triggers division, so that afterwards we have this kind of graph. Notice we have a brand new model running in here. Okay, this is the result of what I just showed you. So we have the multi-cell physics running in the environment. We're not seeing the fields diffusing, but I can, inside of the cells, it's expressing the RNA and the protein, same thing that we've been building up from the very start. Because there's a stochastic process, you can see there's some heterogeneity in its expression. You see the cell bodies moving apart, divisions triggering these cells to divide. So here, how many cells do we have here? Maybe a dozen or so. So you can imagine that this becomes a fairly large hierarchy with many different processes operating inside of it. Okay, let's step it up a notch. We're going to increase some complexity. As we were developing Vivarium, we realized it could be a tool to connect existing simulation platforms. It is not like if we build interfaces to them, we wanted to see that they could connect. So we set out to use as many off-the-shelf simulators as we could in building this next demo. Ideally, we don't have to reinvent the wheel. If someone has created a method and provides a nice interface to it, we're going to use it. Here we're using Cobra FBA process. We've added a few additional processes to transform it into a dynamic flux balance analysis. But you can see the flux is internal. This is log scale, which is why it looks like straight lines. But you can see that the external species, which there's 299, there's some secretions coming from the cell into the environment. And these are the glucose is being taken up. Again, you can't see this because of the log scale. And overall, the biomass increases. So this is running Cobra. We use this bioscrape package, build a wrapper around it. This came out of Richard Murray's lab in Caltech. It can run in deterministic mode, stochastic. It's a SBML type simulator, so interchangeable with Copasi or libRoadrunner. Our next processes are this multi-body you saw before. This is actually a video game physics engine that I found online called PyMonk that I put inside of here. And we have a simple diffusion. You can see here there's kind of this great discrete lattice-based diffusion running together. These come bundled as a composite. They work well together, so you can pull them together and connect them. Here is the full composition I'm about to show you. So we have the Cobra processes here in orange, bioscrape, the ODE in green. Here's the external environment. We have a few additional adapters. I'll show you only one of them. So we need here, this ODE simulator reads just individual species. We need to connect them to an external field, which is on a grid. So we have this adapter that reads the cell's location, it reads the external grid, and it updates the species that are ODECs based on where it is on the grid. Okay, and the result of this hybridization, here you can see really all of the frameworks that I just introduced playing together, and you get emergent effects. There's things here that you can't do with individual frameworks on their own. It's the flux balance analysis is driving the growth. So we get here, you can see the total colony mass growing. Kind of this looks like flux balance analysis. But when it's connected with the division process and the multi-cell framework, that translates into this colony growing and dividing. We have some stochastic gene expression. You see here the RNA and the protein lactose permease. This is the lac operon, by the way. As the lactose permease is stochastically expressed, it lets the cells take up lactose from the environment. So you can see here, lactose starts to get taken up. Of course, the spatiality of this of the environment means that different cells experience different external states. You can see over here on the right that at the periphery of this colony, cells are taking up more glucose. That's because there's more glucose available as it diffuses from outside. You can also see some interesting consequences of the stochastic gene expression. Some of these cells have zero of the lactose permease, which lets them take up lactose, and so you see that they're not taking up any lactose, and that translates to very heterogeneous growth rates across the population. I could talk about this figure for a lot longer, but we're gonna move on. Okay, the next step we're gonna just increasingly crank up the complexity. After demonstrating Vivarium in these initial use cases, several people, I'm highlighting three undergraduates that worked with me to help me translate it, all now graduate students in Stanford and MIT. They spent a summer, which ended up being more than the summer, ended up being a couple of years, and we took the E. coli model and transformed it entirely into Vivarium. Here you can see the wiring diagram on the left. We have about 12 different detailed submodels. Zooming up on a piece of it, you see the chromosome replication and how it connects to these different unique molecular states. Chromosome replication is kind of agent-based model-like. In here, we also have, at the core is this metabolism flux balance analysis model. We have ODEs, we have stochastic algorithms running in here. This is, by the way, the E. coli model that we mentioned earlier. You could see Marcus Covert's GLIMPRINT seminar to see how the consequences of this model. It's very large, 19,000 parameters, all come from EcoPsych, and we're working on completing it. Here's just some of the different ways of looking at the simulation outputs. We can cluster the different molecule types into a few categories and see how they're doubling and together, and approximately the correct amount of doubling time. Looking at measurements versus simulation in the proteome, the distribution of different categories of proteins, flux rates measured versus simulated, and all kinds of cell properties that we can derive from the model. Because we had it in Vivarium, we thought, hey, we have this pluggable thing. Let's put it into the multiscale, the environment simulator that I'd introduced before. Something quite interesting about process biograph is that any biograph can itself be made into a process. There's a way to convert a structure like this into a process by exposing the key variables. Here we wanted the E. coli model to interact with the external fields, so we just expose some and can run it as a massive parallel simulation. This ran on Google Cloud. We got a node with 256 cores on it, and had the E. coli whole cell model run as a colony in parallel. So you see the cell over here, that is an E. coli whole cell model with 19,000 parameters. As it grows and divides, we have a lot of massive simulation. If anyone knows of a model in systems biology that's larger than this, please let me know. I haven't found one yet. I think I'm going to claim this is the largest simulation in systems biology. Prove me wrong. I'd love to be proved wrong. We can see heterogeneity across the population. Some proteins are not expressed at all, and there's consequences to this that we're going to see. Okay, let's some videos. This is the E. coli whole cell colony growing on glucose. You'll see the glucose start to get eaten up. In the same simulation, we can tag proteins. Really easy to do virtually. Here just two different proteins are being fluorescently tagged. I'm joking about fluorescent tag. We're just reading the data. So you can see again heterogeneity in expression as a consequence of this simulation. Some proteins much more heterogeneity than others. Next, we can start to add additional processes on top of it. I'm showing kind of a reduced graphic, but this is the entire whole cell model, just more compact. And here we have a few additional modules that we added on top of the whole cell model so that we could simulate response to antibiotic. We have an antibiotic transport, antibiotic binding, and cell wall growth that is described in this paper over here. This is in response to tetracycline. So you're about to see the antibiotics. There's the antibiotics were introduced into the model and into the simulation. Gray cells have stopped growing, and you can see that because of the heterogeneity in the population, some cells are still growing because they have the correct proteins expressed. Another case of missing function. Can anyone find what's wrong with this image? I love you have to think outside the box a little bit. Here we're expressing flagella in this image. You can see every cell has some amount of flagella. Some have more, some have less. But what's wrong? So what's wrong? If they have flagella, they should be moving. We're just counting flagella here. We don't give them function. There's something very wrong with this image. This should not. This is not how it would be in reality. So we need to add function. We can't just count flagella. We need to give them a model that describes what they do so that they can move. And that's what we did in this paper over here. We added a few processes. One of them, a chemoreceptor. This is a statistical mechanics model, the Monod-Wyman-Shango model of chemoreceptor activity. There is a signal cascade here that I'm not showing, but controls flagella activity. Flagella activity was done with a stochastic switching model. And you can see that when we add these modules to the cell, well, first, if we add just the flagella activity, just the motor, then you get motility, this kind of random movement. But if you add a receptor that is connected appropriately to the flagella activity, you actually get chemotaxis, and you see that these blue lines are moving up the ligand gradient. This is a bit of a break in my talk right now. I have the next slide is actually gonna switch to a demo of Vivarium 2.0.
I had a question.
Yeah.
So there was an article in Cell that got a lot of hype about creating a virtual cell, basically using a bunch of AI models. And the conclusions were very like, yeah, we can solve all these things, except we're going to have to sacrifice some mechanistic information. In the future, do you see yourself adding any like machine learning models to Vivarium, or are you sticking with an entirely mechanistic model?
Great question. Yes. A lot of people see the rise of AI and think it somehow threatens mechanistic models, like it's going to replace, it's one or the other. Only one will survive. I don't see, I think that they're actually very complementary approaches. Whereas mechanistic models are explainable and compact, machine learning can capture unknown mechanisms, arbitrary functions, and also surrogates. So absolutely, I plan to integrate them. These modules could, let's take a look at this one. Any of these could in principle be a neural network. You can the fact that these are mechanistic models is, I think, just how we've been working on it so far. So we could replace any of these with machine learning modules. You could actually take the whole thing, like how I showed you this. Okay, over here, we can take the whole simulation, describe some inputs and outputs, expose them, and train a surrogate on those inputs and outputs, and then replace the model with a surrogate. Probably be faster. But yes, I think there's actually a really nice complement. One thing that's really complementary between machine learning and this kind of framework is this exposing of variables, describing the inputs and outputs, exposing them. That's exactly the kind of training data that machine learning needs. So to answer your question, I think there's many ways to connect machine learning within this kind of framework. All right, let's talk about Vivarium 2.0. So for the next generation of the software, we're trying to make it more general and more standardized. We have a few different interlinked libraries that separate out issues related to running these simulations. We have this schema library that checks all the types, knows how to serialize your simulations, kind of handles the underlying structure, does static checks on the structure. Everything is now based in the standard exchange format, so you can at any point in time take the whole simulation, save it to a file, a JSON file, and then bring it back from that point on. This helps with sharing models, save points, and bigraph schema helps with that serialization. The next layer on top of it is process bigraphs. This makes it this adds the actual processes that let it run like a simulation. We have a visualization library that knows how to read this standard format and create graphs out of it. And then finally, this is kind of a new top-level interface that brings them all together. It's a Python API that, without having to write the documents directly, you can specify simulations, run them, visualize them. They're available on the Python package index. Everything that you're about to see is under heavy development. And you're free to use it. It's all open source, but you might want to contact me. And please do. You're welcome to contact me if you want to use this. Okay. I'm also so before I click on this and go into the demo, I want to do the acknowledgement, since I might not come back to the slideshow. I have a growing lab that's wonderful to have around here in front of the Center for Cell Analysis and Modeling. I've been blessed with a lot of recent funding that's very exciting, and we're always looking for new postdocs and engineers to join the team, so you can contact me at this email. All right, let's open up this notebook. This is like our most developed demo of Vivarium 2.0. You can find it in our GitHub under Spatio Flux, and this is still evolving. So if you see this online, you come here later, you'll probably see something quite different by the time that you visit. Okay, so I want to show you kind of a workflow of how we build these models. I have I'm importing from this library a vivarium that has a few processes preloaded into it and a few different data types preloaded into it. Think of this as like a workspace to build your models, and I've imported things so that they're ready to go. This is all in Python, and there's work to do the same thing in different languages too. Okay, so here we make a fresh vivarium. It just no structure yet, but we have all the types built in. You can view the types. There's 101 types in here, 102. No, sorry, 101. All kinds of different units. You'd see also in here things like more complex data structures, NumPy arrays. Here's the processes that are loaded into this workspace. I have what you'll see that we'll have this dynamic flux balance analysis using Cobra, diffusion advection, minimal particle model, and then this is for a kind of a particle-based engine. So we might want to inspect how to configure one of these. We're always looking to make this easier. Not all the way there yet, but in order to make a dynamic flux balance analysis, you need to give it a model file. This is an SBML flux balance analysis type. You need to give it some kinetic parameters. This is a mapping, and you can override the built-in bounds. So here we're just changing the bounds on oxygen exchange and ATP maintenance. Okay, make a fresh vivarium. Here, using the API, you can just add a process like that. No need to write in pure JSON. You can see as a result of adding this process, we now have this disconnected DFBA process. Okay, so we want to connect it. This is invalid. We can't run a disconnected process. So this if we would try to run it, it wouldn't let you. We're going to connect it. So we're going to add some objects for glucose, acetate, and biomass. Here for each of these molecules, we're going to add an object and just assign it a random value. And then we connect this DFBA process that we created. We're going to connect it to each of the molecules. So you can see how that's structured now. So this DFBA process is going to be, it has a lot of internal state from that SBML model file that I put in there, but now it can read the external glucose, acetate, and its own biomass. We can update the values as needed, get the values. Okay, so this is how we're starting it off. We can save the file at this point. This is showing you what the standard file format was created under the hood. This is the stuff that I'm trying to save you from having to look at and write, but it's there. Okay, load it back in. We're making a new vivarium. This is just to demonstrate you can always save, reload. I could send you this JSON across the internet, and if you have the DFBA process that's specified in here, then you could run it. Right now, this is a local one. We could, in principle, point somewhere online, HTTP, and then you won't even have to have it on your own computer. Okay, so now we have this one. We can run it for 60 time units, get the results. We have some generic plotting. So we're just saying plot these three fields together. You can see the glucose being eaten up. At first, acetate is secreted as the cells grow, but when the glucose runs out, the cells shift and start to eat the acetate. And so you can see the acetate. And once everything runs out, the biomass stops growing. Okay, we're going to crank it up again. I think you'll hopefully starting to get my my approach of increasing complexity. So now we're going to do this simulation, but on a grid. In this case, a 3x2. So we're creating a NumPy array, random NumPy arrays for our 3 molecules, 3x2 arrays. And then we put a process in each one of them. So you can see we're going to make a DFBA for each row and each column. We're going to add it in there, and we're going to connect it to that row and column in the array. So here's the consequence of that. We have our 6 DFBAs. These are NumPy arrays. I'm not showing they're like reaching into the NumPy arrays and connecting to the specific location through the wires specified here. We can change the values, get the values. Okay, so now you can see it's an array. The glucose is a 2x3 array. The acetate is a 2x3 array. The biomass is a 2x3 array. Run the simulation. Here I just kind of cluster them by location, so you can see the 0, 0, 0, 1, etc. We can also plot them like this. Plot snapshots. So you can see this one starts off with a high amount of glucose because I set it, I believe. Yeah, I set the glucose high over here. Okay, so we start off with a high amount of glucose. It gets eaten up. The acetate gets secreted and then also gets eaten up. You can see the biomass increasing, stabilizing. Okay. Next. Here we have our diffusion advection process. I'm going to make a 10x10 grid of it. Here we make a fresh new vivarium, so we're starting over. We add our fields, 10x10 random fields. We're going to add this diffusion advection process, connecting it to the fields that we generated. So we have this kind of thing. You can see how it's connecting at the top. It's going to read these fields and diffuse them. Okay, so we run it. There's the result. You can see that this one actually has some advection. And let's see, because biomass has advection. The other ones don't have advection going on. Okay, so biomass, you can see it's slightly kind of advecting upwards. Okay, next. I hope everyone's following along here. Please, anyone interrupt. I love interruptions. Okay, so now what we're gonna do is we're reproducing this method that was invented in Daniel Segre's lab in Boston University called COMETS. It's essentially dynamic FBA plus the spatial diffusion. And so what we're going to do is take just those kind of small modules, assemble them together, and recreate this modeling framework in Vivarium. Okay, glucose, acetate, and biomass. I'm doing something a little bit different here. So we're going to set the glucose a random value between 0 and 10. The acetate's going to be zeros. The biomass field is going to be zeros except for a little bit of biomass that I'm putting in a location at the top. Okay, we add that to our new vivarium. Here we're going to add our diffusion advection process that you saw before. And now we're going to add a dynamic FBA at every location. So it's going to both be diffusing around and dynamic flux balance analysis is going to be operating at every location. So we go through our rows and columns, add the process, connect it up. So we have something like this. I'm actually not showing you all of them because you wouldn't be able to see it. So we're showing the I guess this is the 19, row number 19. This simulation has 200 bins, so 200 DFBA instances, and one diffusion advection process to link them up. And here's the result of that. You can see the biomass that I started off over here. There it is. It's starting to eat the glucose and secrete acetate. This is not realistic diffusion rates, by the way, but the individual models are the, you know, a proper E. coli SBML model. There we go. Okay. I'm just going to keep on going until someone interrupts me. So I hope you're enjoying this. Okay, we can look at specific bins within this simulation. You can see like site 00. Okay, next up, we're gonna add some particles into the mix. Okay, we create a fresh vivarium. I wanted to look at what's required to configure particles. It requires bin, the number of bins, the bounds, diffusion rate, advection rate, the probability of adding new particles.
Can I actually ask
Yes.
a question?
Yes.
So how do you control for error propagation through all of these models? And what methods are you actually using, for example, for diffusion and advection to solve them?
Yeah, at present, we're not controlling for error propagation. That's kind of another level above this simulation, which we do have work to add. Error propagation has to do with the relationship between some observed data measurements and the results that I'm showing you. So we would have like one additional level, maybe like could be put into a module on top that reads it, and also, you know, is checking the doing some additional processing to check. Can also have another for there is error could result from the time stepping, and so you could have an additional process that controls the time steps to reduce the amount of error. The diffusion advection process that we're using here is how to describe it. It's just based on a lattice space. It's as simple as you can imagine.
It's okay. I'm a mathematician, so if you want to say the exact method, like is it like upwind or?
Yeah, the name of it is slipping. It's a discrete space diffusion method that you essentially calculate the difference between
Finite difference?
Finite difference.
And is it like the second order, first order?
First order.
Okay.
Simple as possible. But you know, in principle, we could add new modules in there that are of additional complexity and accuracy.
Okay. And then, so you're basically using like some type of operator splitting because you're not solving it all at once. And so how do you control, so, you know, 19,000 variables, that's a lot. So you acquire a rather large operator splitting error.
I think that there's certainly a lot of error.
Okay.
Yeah. With the E. coli whole cell model, we adjust the time step, but I do think that we need to look at that in greater detail and control it. The E. coli model, you know, we just compared it to the data that we know. And, you know, I showed you some proteome comparisons, fluxome comparisons, and so that makes us feel pretty good. But I think there's a whole research, you know, domain that we should embrace and bring in and build into the Vivarium framework. I should also mention that anything that applies here to this framework applies to other agent-based models as well. This is just kind of like generalizing a lot of agent-based modeling approaches, but the same problems are inherited.
Thank you.
Thank you. Okay, so here we have particles. This process, I'm just initializing it with 2 particles. So you can see that each particle has this ID. You can think of it like an agent. It has a location, a size, a mass. This particle movement process does the Brownian motion to move them around. So it operates on all the particles. As I was saying, we're going to add particles to the top of this simulation. Okay, save it, run it, plot it. Okay, particles are there's some downward movement. I called it advection here, but I know it's not actually advection, and so we'll be fixing that. Here you can see the end state after I've run it. A lot more particles have been added. Okay, now we are going to put the particles with the diffusing field. And we're going to put an additional minimal particle process inside of each particle. So now each process inside of it, there's going to be a process that reads the external field and grows based on glucose and acetate defined here. This one's not rendering correctly, so hopefully by the time you check this, all the online viewers, this will be a process. This minimal particle process lives inside of this particle, and you can see diffusion advection operating on the fields, and you can see the particle movement also. Okay, so we have particles moving around and diffusing fields, and they're uptaking and secreting. I see here that this is negative, which is not good, but hopefully you see the potential for building these large-scale hybrid simulations. So at this point, I could export this like a template. People could adjust the parameters, replace the processes, and it's reproducible because everything is specified in a file underneath. And I think this one is maybe not complete yet. Yeah. Okay, so the final demo, let's see, does it run? No, okay. So that's the end of the demo.
Sure. I would like to inquire a little bit more about how enzyme production is modeled and how it ends up getting coupled to reaction fluxes.
Oh, yes. Okay. I have a few examples of that. So it happens in the whole cell model, but it also happens here. Okay, so here we have a few enzymes. This lactose permease is an enzyme. It's being expressed. We use Michaelis-Menten to calculate a flux based on the enzyme concentration and the substrates, Kcat, Km. Once you have that flux, it becomes a constraint on the flux balance analysis model. So we have in flux balance analysis, you might be familiar with this, but you define the upper bounds and the lower bounds, and so we take the the flux calculated by Michaelis-Menten just set the upper bound. So it could be less than that. But typically FBA, since does the maximization, it goes all the way up to that maximum allowed flux. Here we're doing it for at the boundary, essentially the exchange fluxes based on using this permease. In the E. coli whole cell model, in here, this metabolism process that you can't see, I know you can't see it, it reads in enzymes, and I think it's about, Arnab, who might be in the audience, knows the number, but I think it's about 300 enzymes are kinetically constrained. Arnab, are you there?
Yeah, it's around 300.
Around 300, okay.
309, I think.
There you go. So 309 of the enzymes are kinetically constrained. We have our transcription, translation, complexation processes generating those 309 enzymes. The fluxes are calculated based on their concentrations and substrate concentrations, and then we set the bounds of those fluxes in the model. Those are, whereas the previous one was mostly on the boundary using exchange fluxes. For the E. coli model, these are mostly in core metabolism being constrained. And we're kind of working, so you can start from the outside and work in, or you can start from the core and kind of work out adding kinetics. Based on what was available to us on EcoPsych. So clearly we don't have full coverage at all.
Okay, thank you for clarifying that. And would it be possible, for instance, if you don't have data governing, say, gene expression, to still model the kinetics in here?
Sure. So you can gene expression will get us the enzyme concentrations. If we have I don't have a Michaelis-Menten equation handy, but you can take the kcat times the concentration and replace it with just a Vmax, which essentially, so no longer is the enzyme concentration included. You're just assuming like some constant enzyme concentration with the Vmax, and then you can do it with just the substrate concentrations, which I think is actually more common because most dynamic FBA models don't have gene expression to go along with them.
Okay, thank you.
Good question. Yeah, that's an interesting way of connecting flux balance analysis with kinetic, that kind of dynamic models.
So I have a question about orchestration. And so I guess one of the benefits of using this graph rewriting approach is the fact that when you do have to apply a rule, you can apply a rule locally in the graph. And so you don't have to take into account all the information. And so how can you provide some more intuition about how to connect this ability to apply rules locally in this multi-scale context with respect to the scheduling that's done in the software to kind of prioritize which event should be triggered with respect to different timescales?
I'm thinking about how to describe this in a way that won't lose too many people. So we have the orchestration engine. We pass in the composite definition in, and it takes over and does the orchestration at that point. If you have these temporal type processes, then they do multi-time stepping. Okay, the reaction rule, the graph rewrite reactions are done in the way I described here. They're done between time steps. All the ones that I've used happen between time steps. For the most part, the ones I've shown, there's some kind of threshold that happens. So let's say we have these processes update, and we recalculate the total mass of the cell, and then we have a divide process that has a threshold. It says when the mass crosses over this threshold, you're going to trigger the rewrite rule. And so between every time step, it evaluates, evaluates pass. Now the threshold has been crossed. It's time for it to send its graph rewrite rule up. So here, and this is the thing that I failed to include when I was making this last night. There would be like a division process here. Imagine a rectangle down over here. That's just, let's say it's reading the volume and it's waiting for that volume to grow. As soon as that happens, it passes an update to the cells telling, so a local graph rewrite rule that says cells, you have to divide cell 0, and it tells it how to do it. So it just those rules get passed, the graph rewrite rules get triggered as events, just based on where their wires are pointing. I hope that was clear. I think we could talk about this for a while.
Other questions or discussion?
I have a question.
Go ahead.
Have you guys considered like low code visual solutions, like visual programming on top of your framework for the creation of these models? I could see something like Total.js Flow being potentially something you could integrate into what you already have.
Great question. You can clearly imagine this kind of interface. Like I almost, as I'm writing these things, I want to just drag and drop. I want to like drag on the arrows and pull them around. And the answer is yes, we actually have started that. Let's see, is Alex in the audience? Alex Patry, are you there, Alex Patry? No, I think he might have logged off. He was there earlier. So we do have an early prototype of exactly that kind of framework, where you have the available processes, instead of listing them like so if we go to the demo that I showed you, instead of just listing them as a list, you can imagine like boxes that are now available to be pulled in and wired together. It can do the type checking for you. So if you try to connect to the wrong thing, it's not allowed. It'll give you that kind of feedback. The end result of any kind of API, such as the one that I'm describing and the one I'm showing you here, is it needs to be able to compile down to a document like this one. So we've actually had a number of different APIs being defined recently. Only one of them a visual API, as you had asked, and other just kind of scripting APIs. But there's different ways to do this. I think the visual API would pull a lot of people in and make it much more accessible.
Awesome, thank you.
Absolutely.
I have a question. Is there a way to validate or calibrate your parameters in Vivarium?
Okay, so the answer to that is it has to do with the module. We can pull in different modules. So let's say like this one over here for the ODE stochastic process that goes into here. This one has some built-in parameter estimation within it. So we just leverage those existing methods. The other, there's a more difficult question of like the whole thing together. So we could calibrate the individual modules and feel pretty confident about them, but then when we put them together,
yes,
know about that. And so early days again, but it goes back to this point over here that I think I actually have in my appendix. I have it a little bit better. So let's think about this is a composite process. You can if you have a full composite simulation, you can always transform it into a process and expose some inputs and outputs. And now that is a module that you could put in some state, read out what the prediction. Of course, the parameter, if you want to adjust parameters, the parameters are passed into individual processes. So it's a little bit more complicated. But in principle, you can expose the parameters, read the state out, make adjustments. We've done parameter scans that way. So we can just do the parameter scan and then see the regions in the parameter space that reduce some error. But as for like more principled approaches to do it, like I think there's still some open questions.
And is there a limit number for the number of particles that you can consider, or is just the time or the size of your computer?
It's the time and size of the computer. So we've actually did some very deep profiling. This is horizontally scalable. Things run in parallel. So if you add new processes, it's a parallel. And so it's really limited just by how much compute you have. There's also another potential bottleneck is communication. So if you have really high communication between your modules and you add more modules, that can start to add up, but there's strategies to reduce the communication overhead. You can put some processes local and share memory, some distributed, yeah, overall, this is a very scalable framework.
And now a very, very speculative question, right?
Mm-hmm.
Is it possible that in the future you'd have something that the program would change its parameters or even some of the things and then evolve by itself?
Oh yeah. So this is some science fiction, but I think we can get there absolutely. So we can already with a coli model, when it divides, we can perturb the parameters.
Yeah.
We actually have is Tasnif in the audience? Tasnif is a postdoc in my lab. He's in the audience, but he's working on when you launch new cell models with some mutation. Some will grow, some won't grow.
Yeah.
We can let that evolve. We can let it evolve.
Yes.
In a way that this model. Let me show you back to this simulation. So this is the antibiotic response. As soon as the antibiotics are introduced, there's cells that stop growing and dividing, and the cells that are growing and dividing, there's selection pressure, and you can we actually did a phylogenetic analysis of these, that the protein expression rate is going to go up. So there are signs of very of this
yes.
is possible. Yes.
Yeah. Thank you for the very nice talk.
You're all welcome. Okay.
Well, we've had a good discussion. Was there a question in the chat as the last question before we break?
Okay, so we have okay, Mohammed. I have a question for Mohammed. If I missed some questions, I apologize. But okay, is there any work done using this software for fibroblast cell migration, haptotaxis, chemotaxis, protrusion, retraction? Not all of that, no. But let me show you one thing I was anticipating. Here we have a this is a model done with John Hickey of tumor T cell model. You now are all experts in this visual language, so you understand this perfectly. Here we have this is actually comes from data, the initial conditions we have from data, and we can run this and simulate kind of tumor growth and T cell interactions. Not exactly what you were looking for, Mohammed, but this is about as close as I have for it.
Okay, well, I want to thank you very much again, Eran, for the demo and the talk. It's a very exciting project. I think a lot of us will use it. And I want to thank everyone for coming today for the new time. And Eran, I hope you'll come back in the not too distant future and give us an update on your progress. Thanks, everybody, and we'll see you in 2 weeks.